Hi, everybody. Welcome back to Psychedelics Today. Joe Moore here, joined by Dr. Rochelle Hines and Dr. Dustin Hines at UNLV, also with Tesselate, website tesselate.ai, TSL8.ai. Is that right?
Yes.
Perfect. Both of you are amazing Canadian neuroscientists. I'm really excited to dig in. And but you're living in the beautiful city of Las Vegas, which we had a conversation about. It's far more southwest than anybody gives it credit for. It's not just the Strip.
Yeah, I think we've been to the Strip 3 times. It's amazing, but it's just not our vibe.
Right.
The more desert people.
Yeah. And the access to the desert is endless. I've been wanting to go to Red Rocks to go rock climbing for ages, and it's just right there.
Good.
So I guess let's do a super duper high level of what is Tesselate. Then I want to dig into you both as individuals. Who wants to kind of describe what Tesselate is?
I think the first thing we should do is probably get into what is a tessellation.
Yeah,
and
a good call.
Tessellation really is. Sorry, go ahead.
That's a good call. Let's do it.
I think a tessellation is really one of these captivating patterns where kind of all the shapes fit together perfectly. Really, our idea, having a company called Tesselate, comes from our time in industry where people didn't look at the shapes of the receptors. They didn't look at how the different areas of the brain work together. And what we really are trying to do at Tesselate is think about all the neurotransmitters and all of the person's problems at once to create kind of a holistic approach to therapeutics. So tessellation really is trying to get what we call polypharma drugs that can work together to affect, mental health, the mental health crisis.
I love that. Yeah, we had a long, great conversation the other day about how pharma is like really simplistic and the body's a little bit more complex, but y'all are right at the tip of the sphere of this kind of tide shift, it seems. Like, okay, sure, biology happens and we need to do that.
For sure. I would blame neuroscience too, which we're firmly part of. Neuroscience has been so reductionistic, since it's ongoing, which we need it to be, looking at a single synapse and a single receptor. But it turns out your brain has billions of receptors and billions of cells connecting. And really, that's one of the thrusts of the company too, is to understand some of these basic connections.
Mm-hmm. Absolutely. Yeah. And now, what are you all looking at then, specifically, or what was the origin story?
Well, I think the origin story for us is really Dustin and I fit well together too. And, we really had complementary skill sets and complementary ideas about what we wanted to advance. And, we've been sort of on a quest our whole careers for novel therapeutics. And when we came across the progress being made with psychedelics, we were sort of truthfully captivated. We were just almost dumbfounded at the level of effectiveness. And so, that...
When was that roughly that came on your radar?
Yeah, I don't know. When do you think, Dustin?
2011, I think.
Mm-hmm.
We were screening lots of compounds at that time for depression. And if we had a compound that even showed a 5 or 10% change, we were pretty excited in some basic tasks, both in DISH and some whole preclinical models like the 4-swim task. And, we tried some of these ligands and they were like 80% change. And we didn't believe it at first. We were gobsmacked and we thought this is something we should really look more into.
Right. That makes a lot of sense. So yeah, how do we even describe that? So a lot of people right now are kind of freaking out on, psychedelic science not being legitimate or something like that. And there's a lot of hype and, kind of fair, kind of not. There's a lot of really hard-nosed scientists looking at this data and trying to make the best, data they can. Right. So if traditional pharma is looking at, 5% gain, 5% boost, and that's amazing. Does the FDA think that's amazing? Especially when we look at this, because there has to be a substantial benefit over competing drugs for the FDA to approve it. Right. Is that somewhat fair? Yeah. So how do we go to that next stage of okay, look, this thing maybe is 30 to 70%, versus a 5%. Why wouldn't we dump a whole bunch of cash into that as a biotech? How do you all think about that?
I completely agree with you. Why don't we? But I think there has been this idea, one pill that fits everything, right? So we're really coming out of this age where every kind of pharmaceutical was guided towards curing a 70-kilogram white male of his, current ailment. But turns out we're a diverse population. Number one, not everyone's a male, surprise. And then, there's a lot of different kind of effects with different groups of drugs. So we're really trying to get into this personalized aspect of medicine. So I think the change is here. I think there's a big renaissance, we know, in the psychedelic field now, but there is a big renaissance in neuroscience, in medicine. We're really looking for innovations that we haven't truthfully had in 50 years.
That's a big deal. So I just recently was made aware of like issues in medical devices, even like the pulse oximeter, is that what you call it, that little finger device? And like people of color, it works very differently in many cases. And like how many bad health outcomes have come from that? And now we understand there's biodiversity in human populations. We've got to really address that, if biodiversity is the right term.
Yeah. And depression itself is extremely heterogeneous. So if we take a population of people with depression, they have all kinds of different features that are extremely important to consider. And so, it makes sense that one type of medication isn't going to work for every single one of them. People with depression may have more of a complement of anxiety or more situational and environmental or trauma-based root causes. And other individuals have, maybe more of a biological or genetic or family-based root for their depression. And so, of course, each of those people is not going to respond the same way to the same drug under the same conditions.
Exactly.
Yeah, and I think part of the problem too, it is a huge useful tool, but the DSM is kind of a catch-all. You're trying to put everyone into a big category so then you can diagnose. But the problem with that is you kind of miss a lot of things. You put people in categories they shouldn't be in, and then some people don't get into the category that they need to be in. And so there's this huge move to neuropsychiatric or psychiatric personalized medicine. And I think that's the age we're in. And I think psychedelics fit into this really well.
So we haven't done too much on that topic, but what's the general thesis? Are we looking at genome plus like gut biome plus personal history plus a few other markers?
Yeah, so we're really interested in some of those peripheral biomarkers that can be attained relatively easily. And so there's getting to be a good amount of data about subtypes of depression that feature certain biomarkers, maybe related to inflammation or immune activation. And then there's other sorts of things that we're interested in looking at. And then the other really big component for us is actually looking at EEG. And EEG we can do in our preclinical models. It's highly translatable. So this is recording of the electrical activity of the brain. And we've found EEG signatures that we think are really helpful in predicting the outcome of the therapeutic session. And so Dustin can maybe talk more on that too.
Yeah, we published a paper about a year ago now, really looking at this problem that when you look at screening for psychedelics, if you have a highly potent, highly selective psychedelic and you use the canonical way to screen for psychedelics, which is the rodent head twitch, you don't see anything. And so we really came at this with saying, well, there has to be markers. If you have a drug that's really amazing and really selective, there has to be something that changes. So we found this signature that we call phase 1. And it's a beautiful waveform, at least beautiful for nerds like me that like brainwaves. And it's this delta and theta wave kind of riding on each other as the whole cortex beautifully starts talking to itself. And so, we can use this phase 1 and see that it scales with dose. So the more selective doses we get, the head twitch and phase 2, as we call it, might fall off, but we get more of this phase 1. So it gives us this elegant way to screen for compounds.
That's super interesting. And how effective would you rate head twitch alone, like traditional head twitch?
Well, depends on the drug. And that's the one thing, again, with this new screening method we've been able to find. I think we try to come up with these techniques as scientists, like the head twitch, and then we try to broadly apply them. But, if you were to talk to any psychonaut and say, is DMT the same as psilocybin? Absolutely nobody would say they're the same, but we would screen both with a head twitch. They're completely different. So I think at high dose and different drugs, it's not reliable. I think if you can hit the right dose instantly with, say, a novel compound, it could be effective. And again, with Tesselate, we still use that as kind of a benchmark. to see if it does produce, the novel compounds do produce head twitch. But when we find the highly therapeutic and highly selective compounds, there's much less head twitch. And again, a much higher therapeutic output, which are things like increased spines or synaptic connections.
Mm. Right. There's like a whole host of things you can look at. And
Absolutely.
that's got to be
And we
kind of
are.
eye-opening. And you are. So like, what was that moment like when you realized, oh, holy crap, look, we can get way better performance this way from our research, in terms of like output per hour and dollar of research?
Yeah.
Yeah.
It was, for me, I would call these paradigm shifts or eureka moments. It was a huge eureka moment. So we were giving pretty high doses of LSD and 2,5-MeO-H, which is an NBO. And what we noticed was there was no head twitch. So me looking at the mice and having lots of training in kind of like behavioral neuroscience, I could see these mice were not on this celestial plane. They were like staring at the ceiling. So what we looked then was at the EEG. And when we saw the EEG, we saw these beautiful waves that, weren't akin to, but were similar to what we see during rapid eye movement or REM sleep, these big, big sawtooth waves. And then they were episodic. So as the animal would kind of come back into reality, whatever that means, we would see that the waveforms would go away. And then as it would start to trip again, we would see these big, big theta delta waves coming through. And it was like, wow. And then when we changed dose a couple times and then added nicotine and some other compounds, we even got further insights into kind of how this phase 1 and phase 2 work with each other.
That's excellent. Wow. Okay. Now, I guess let's rewind the tape. What made you interested in neuroscience to begin with? Like, I assume you got exposed to something as an undergrad, but you never know.
Yeah, I guess I could go first. I was actually working in education. I was thinking I was going to become a teacher. I was working in special education, working with kids with autism, actually. And I thought, maybe if I took a class on the brain, I could do a better job. I could understand the kids better. And so I picked up this class as an elective called Brain and Behavior. And literally that first week in that class, I was like, this is it. Like, I switched my major. I started working in the lab of my professor. And, prior to that, I wasn't aware of neuroscience as a field. And, I was always sort of sciencey as a kid. I had a microscope, I had a rock collection, true nerd through and through. But, it just hadn't occurred to me to pursue science literally until I was sitting in that class. And I was like, this is amazing. Like, I was just completely captivated.
Like the fact that there was like physical correlates to behavior, things like that you could see. Yeah, totally. And how about you, Dustin?
So kind of a similar approach in my undergraduate. I was kind of slated to do philosophy and astrophysics. I was really into physics and math. And all these waveforms I was looking at the time were pretty cool. And then I started working in the lab of Dr. Ian Wishaw and looking at brain waves and animal behavior. And again, it just grabbed me right away, where you have this pure sine wave, right, that you can predict. You look at the EEG of an animal, you have no idea what's happening next. And the fruit for us to understand the brain will never end, right? It's understanding ourselves and understanding science. And I'm not kind of trying to diss physics, but there's some pretty big formulas that have been solved. And there's nothing really solved, in my opinion, in the brain. We're just starting to understand our brain.
Really just getting there. Like, is there an analogy? Like, a lot of people are saying things like, oh, we figured it all out. We know everything. Like, I'm sure some physicians who are like, oh, we've got it all figured out. Like, we're fine. But is that just part of their like placebo prep for their patients, or what's going on there? And like, where are we actually as far as neuroscience goes? Like,
well, let's talk about
like
one of my
3%
favorite topics lately, which is the cortex.
Right.
And so traditionally, we've looked at the cortex. This is kind of the outer coating of your brain. The front part of the cortex is called the prefrontal cortex. And this is really what chiefly makes humans humans. It's used for planning and sequencing and kind of all these other words that I don't really understand, like executive function. And we've always taught, and I actually taught this morning, that it's layered 1 through 6. But when you look at it, it really isn't. There's a whole bunch of different layers. And most of the interesting things now that we know in the cortex have nothing to do with layers. And in fact, psychedelics are helping us and others to see this, that most of the interesting properties of the cortex are laminar. They're not columnar 1 through 6. That's fascinating to me. That's absolutely fascinating. So where are we at? I think we're just starting over again. We have some good ideas, but we're really just trying to look at those ideas and see what works and what doesn't.
So I'm
There are
familiar with the
other,
idea of a laminar flow hood, but could you describe like laminar in terms of like the neural perspective?
yeah. So really, most of what people think is you have this layer 1 through 6, and they make what are called columns. And each of the columns work in layer 1, 2, 3, and 4, have all typically these stellate cells that help kind of gather information and integrate information. And then the remaining layers are largely pyramidal cells that output. So that's a simple input, integrate, and output circuit. Well, it turns out that different parts of the cortex have different structures within those layers. So for instance, sensory cortex has way more capability to integrate an input. And then things like motor cortex have a lot more capability to output and fire muscles and do things like that. But the big difference is that all these areas are connected in a laminar. So it's not a column. It's instead of that 1, 2, 3, 4, 5, 6, all the neighboring cells kind of work with each other. You can see this most clearly, I think, in the visual cortex, where you look at something called ocular dominance columns. And ocular dominance columns are where the right eye sees all of the right field and the left eye sees all of the left field, but they both kind of see the opposite field also. And so the brain wires up to see what the left part of the world is and the right part of the world is. And this isn't columnar, it's laminar. So really our perception of the visual world from our eyes doesn't fit into this kind of idea of columnar of the layer 1 through 6 at some level also.
Mm, that makes sense.
Yeah, because we're understanding now a big feature of the cortex is to project to itself and to understand what other parts of the cortex are doing. And so that's, really an interesting aspect of the laminar is that, regions talk to one another, they talk to the other hemisphere, they're receiving information from those sites as well. And so, it kind of is interesting to see, when they look at human patients under fMRI using psychedelics, they see this enhanced connectivity between different cortical regions. More parts of the cortex are talking to other parts of the cortex rather than, more input output unidirectional circuit. It's more of this, the cortex speaking to itself, which is really fascinating.
Mm, it's interesting. I was having a conversation with Gould DeLynn from Hopkins not that long ago. Have you seen her work or you're familiar a little bit? Yeah, incredible researcher. I love the attitude she puts out. It's amazing to me. And then this idea of the amygdala being the thing that MDMA is interacting with in particular, and this being the mechanism of action that allows the therapy to work, and that being kind of a little bit simplistic. And she said something similar. She's like, yeah, like when I'm talking about this in class, like totally, I use this analogy. But when I am doing my work, I'm like, oh, this stuff's working at more of a molecular level because octopus don't have those same brain networks. And we diverged well over 100 million years ago. I wish I had the real firm number, but I find that fascinating.
And I actually really believe, again, at this golden age of neuroscience, we're going to figure out that, we kind of have these ganglions like sea anemones, these nerve nets, and also like squid. We don't really understand what they're doing in humans. They're still there, right? We are basically, as humans, if you think about it, a nematode. We're just a worm. We've got a mouth and a derriere, right? A giant tube. And we have all these nerves coming off of that. That's basically what a worm is. We also have these ganglia, like a squid. We have those too. And what we've really only focused on is a single thing, the neuron in a brain. But the brain talks to the gut, talks to the heart, talks to the skeletal system. And we're really in a golden age. We're going to understand how they all talk to each other.
Mm. If you had to give an estimate, is that a year out?
Oh, that's my career out, 20 years out.
Yeah.
I hope 5 or 10 years. We'll see. And again,
things are accelerating like crazy though, too. So it's so hard to predict anything these days.
For sure.
Yeah. I'm reading, The Future is Faster Than You Think right now. And it's like talking about convergences of technology being like wildly accelerating of all fields, from material
Yes.
science to data to what you're up to.
Yeah.
Yeah, we feel that too in our career, that, the premise of that is that we think it's a first order exponential, but it's really a second or third order exponential, and things are just flying. So yeah, it's an exciting time to be a neuroscientist for sure.
I bet. Yeah. So before we get back into your personal stories, any kind of like super duper insightful or glib comments about Neuralink and that kind of like brain tech interface, or is that just ridiculous?
I think it's pretty cute.
I stopped paying attention because there was some comment about how they don't even need neuroscientists. They don't want any feedback from neuroscientists. They've got it all figured out. So I just actually stopped paying attention to it because it just was that made it too much.
It's super helpful when you have everything figured out, right? It saves a lot
Yeah,
of time.
great. Can you give us some insight? That would be awesome.
Oh, great. So, all right, what kind of work were you both doing at UBC when you were doing your PhD work?
Yeah, so a big focus of mine has always been on synapses. So I'm really fascinated by these connections that cells form with one another in the brain, and I'm really fascinated by how they form these partnerships so reliably, and all the right equipment gets there. And generally under typical conditions, we have this really high fidelity system, so that neurotransmitter gets released, and the right receptors are there to receive it, and the signal gets passed on. And so, that always struck me as just this incredibly cosmic process. How do we make it right all the time, hundreds of thousands of times during development? And so that's really what I got interested in first, is brain development and how do synaptic partners find one another, and what are the intricacies of synaptic function. So trying to understand neurotransmitters and receptors at a deeper level, and also trying to understand the ways that we modulate receptors using substances. So it's really was a focus on specialized types of synapses, how they get to be so specialized, and how we can manipulate them.
That's awesome. Dustin.
And so obviously I was also at UBC because we had been married by that point. And what my focus was during my PhD was in the lab of Brian McVicker, was really looking at how cells talk to each other. So again, in neuroscience, we've always had this one synapse talking to the other synapse. But depending on who you talk to, the other cells in the brain outnumber neurons 12 to 1 or 2 to 1 or 3 to 1, depending on how you count them. But the point is there's a lot of other cells other than neurons. One of the chief other types of cells is a glia cell. And these glia cells both talk and listen at the synapse. And I thought, wow, we're trying to modulate the brain and understand brain modulation, and we haven't even looked at 2/3 of the cells that are responsible for that. So I looked at an immune cell that can modulate synapses. It kind of goes in and nibbles away and gets rid of that kind of the unneeded synapses, called cellular pruning or synaptic pruning. And then I also looked at a behavior called sickness behavior, where you feel kind of really blah when you have, the flu. You don't want to get off the couch. You kind of don't want to eat ice cream or do any of those cool things. And it's really a correlate for major depressive disorder. Not that it's the same, but a lot of the underlying conditions are related. And, this was kind of some of the first work looking at the fact that maybe inflammation is a huge player in mental health.
Thank you for that. Yeah, it's there's so much basic science left to do that it's like, industry's making guesses, right? Educated guesses. But yeah, so you two are also doing this startup, Tesselate, and what we've seen in the last number of months might be alarming, but you might also have insight. Like, hey, look, that company just totally crashed and burned. Like, why? And now you can actually learn from them and kind of iterate better. Like, it's just a strange time to be jumping into business, but it's, it's also the time to be jumping into business and building here, because there's not really federal funding for this, and there's so many people suffering that we could help.
Yeah, that is 100% the hypothesis. We easily get 1 or 2 calls a day, Rochelle and I, for people, with dire straits, with severe mental health issues, and nothing's working. And back to that first comment that, it's a tough time to jump in. That's true. There's going to be a lot of companies that fail right now. But I think out of these tough periods, the champions come out, and we really are pitching ourselves as one of those. We really want to change mental health. And I'm sure everyone does, but I think the background in looking at the mistakes of pharma, to go back to what you were saying, it's really expensive to do these trials, and people get really favored in their petri dish, trying to put chemicals on, and then they find out right away when something's going to work with the brain, it probably works on the heart too. And so a lot of trials, are wasted because the toxicology ends up being deleterious to the heart. And so one of the things we're able to do really well is screen in whole animal and understand right away, is this something that actually works on the brain, and is it working on the receptors and targets and circuits that we think it is? So I think people are burning through a lot of money with, molecules that they've spun that really don't have a place in a receptor, that don't really have a place in a circuit, that don't really have place in behavior.
Yeah, and in order for something new to stand out, it really has to be a standout currently. And there's a lot of people pursuing very, very similar types of targets, and it's very difficult to be differentiated, in that way. If everybody's making a different version of psilocybin, I've heard a lot of commentary on that lately, and I think that that's one big challenge is, how to differentiate yourself and truly do something that's different. And going back to what Dustin said, we really developed a strategy of failing fast, because if you can push your leads really hard in preclinical, you save yourself a lot of time and distress trying to push something forward that's not really going to perform, at that superior level. So, that's really what we learned in working with pharma. And, you don't have to take our word for it. There's a JAMA article that says that, the average drug that gets into the clinic costs about $3 billion. And most of that cost is racked up from failures that get pushed along, make it into clinical. A lot of money goes into them, and then they ultimately fail. And so, I think a big strategy now is to see how quickly you can get something to fail, because then you'll save yourself some time and money in the long run. And then, figure out ones that actually do have a chance of performing in the long run.
So this is like where I feel the obligatory, hey, why is biotech worth anything? Because so many people in the psychedelic scene are pushing against it because they think psilocybin's perfect or whatever it is. And, it turns out psilocybin's not perfect. Most drugs aren't perfect. What is even a perfect drug? That's another question. And, for instance, let's look at Denver. Denver decriminalized psilocybin a number of years ago, right? And are all of the veterans healed? Like, no. Veterans have not.
Unfortunately not.
All
that would be awesome.
Right. And it would be amazing if that did work. But what we need is a tight clinical framework with really great data on drugs. And we need a huge network of people to be able to issue treatments and, referrals even. So, we need this muscle of infrastructure of the modern healthcare system, insurance, all that fun stuff, probably a lot of government dollars to be able to manifest the vision we're looking at here. And just mushrooms on their own, great, cool, but it's not adequate. I know people who only want to work with MDMA or only want to work with 5-MeO-DMT when there's so many options and so many new molecules coming down the pipes. Maybe a good comment here is around what is a dirty drug, and are there any dirty drugs in psychedelics? Meaning, I think you know where I'm going with that.
Yeah, so I'm happy to tackle this one since I spend most of my time thinking about receptors as targets. So most psychedelics have action at what we call the 5-HT2A receptor, just in case there are people that are listening to you, Joe, that haven't heard that yet. I feel like 5-HT2A is everywhere these days. Everybody's talking about it. And that is definitely a central target. But we're starting to get some pretty clear information on other receptors that most psychedelics bind to. And some of those may be adding to some of the therapeutic benefit, but some of those may be contributing to negative off-target effects. So, for example, a lot of psychedelics also bind to a very closely related receptor called 5-HT2B. Now, I think the research isn't quite finished here. We don't know for sure if using psilocybin could pose a risk. My hypothesis would be that using psilocybin regularly, even at smaller doses, would be the biggest risk category, because in drugs that have 5-HT2B receptor action that we've looked at in the past, it takes this repeated and sort of chronic use to cause damage to the heart. And so, that the jury is still out on that. We don't know for sure if microdosing psilocybin for a long period of time can activate those receptors sufficiently to cause damage to the heart. One other target, again, is a related receptor for serotonin called 5-HT3. And it's sort of a unique family member. It's a different type of receptor entirely, but it still binds, serotonin still binds it. And it's enriched in the gut, and it's responsible for inducing emesis or vomiting. And so this may be responsible for a lot of the stomach upset that people experience when using different types of psychedelics. And so some of these effects are undesirable and may be barriers for some people who are unsure about using psychedelics as a therapy.
And so I'll put on my lateral thinking hat really quick, because that was excellent. My typical talk track around psychedelics, and this is wildly speculative, so everybody know I'm not citing papers, but I remember Amanda Fielding being on stage at Horizons years ago, and she's really pushing microdosing. When what she was doing was certainly not microdosing back in the day. But she's saying like, hey, look, my heart's totally fine. And I was eating huge amounts of LSD effectively daily for years and years. And look, heart's fine. But that's N of 1, of course. It's not a real study. But where I go is typically like, look at that huge cohort of people who were that 60s counterculture, going to Grateful Dead shows all the time, consuming a fair amount of LSD and psilocybin on the regular, both like dirty molecules, meaning multi-receptor site activity kind of deals. Yeah. And wouldn't we see that? Like, wouldn't we have some sort of cohort data on that? And, no one's done that trial. For me, it would be apparent at this point. I don't think anyone's looked at that.
Yeah, that would be great data to have. And if you look at other, FDA-approved drugs that have been pulled for the same action, like fen-phen, it's not as high a number as you think. It's 10%. And
their dose is massive too. And that's one of the big things I go, like, they're eating huge doses of that when maybe 22 to 20, like 30, I don't know, 10 to 30 milligrams of psilocybin maybe is what's being consumed.
Yeah. So that's not a large number, 10%, really, when you look at how selective that drug was. So I think, like Rochelle said, the jury's out, but it's a worry, right?
Mm-hmm. And we got to get an answer, no doubt. Yeah.
Yes. Yeah. And I guess, a bit our thinking is, if we could avoid that effect and not remove any of the therapeutic benefit, why wouldn't that be awesome?
Is there psychoactive effect from that receptor site specifically that we know of, or is it exclusively a cardiac effect, or we don't know yet?
A little bit debated, let's say that. So there's more information. There's some great studies coming out saying there might be a little bit of effect. But I would say at this time, I'm pretty confident saying the 2A is kind of where the major effect is, but I'm sure somebody will send me.
Do we have any hyper-targeted drugs that only hit that site?
We do.
Sweet.
So
that's great to know.
We have some of our novel compounds are, probably 400 times more potent than psilocybin at hitting that site. So that's kind of what we've engineered and what we've tried to do. And again, these are compounds that would have been missed in traditional screening because the animal just kind of zones out. But when you look at that phase 1 and phase 2, and then more importantly, when you look at the therapeutic effect following, it's huge.
That's great. Now, this idea of Gen 2, Gen 3, whatever, psychedelics, it's like a weird concept for the psychedelic scene to understand. They're like, what's wrong with LSD and MDMA? Well, there's a few things, but it's like, they're pretty sweet, but there are some issues. And I think better predictability and lower dosing are some of the best. Lower dosing meaning like increased safety because you're giving somebody less drugs, right?
Yes.
So, and you're more predictability because you're only hitting one receptor site, right? Like, that's kind of awesome. How should people think about that? Like, the consumer, the investor, just the professionally interested person here.
I think the first thing to say is, again, back to tessellation, we're choosing some super potent psychedelics that are hitting the 2A. But, we have a lot of hypotheses that it's not what happens during this psychedelic experience. It's kind of the synaptic plasticity that comes in the brain following that experience. And so back to dirty drugs, some of the dirty drugs, like the tricyclics for depression, are still some of the best. And so what we're trying to do is kind of take the dirty out of it and add the components that we want. So I think the future really is understanding the brain. And I think the future really is modulating the beneficial effects of psychedelics. And I think psilocybin is a great psychedelic. I don't think it's going to cure every mental health problem. And one of the things in the basic research in the lab that we found recently is something that we've coined the term the trypnogram. And so if you look at all the psychedelics, we're very fortunate Rochelle has a DA license. We have tons of different psychedelics. Turns out, lo and behold, who would have thunk, not all psychedelics are the same. And so if you look at DMT versus, say, mescaline or psilocybin, they're completely different. Well, I think if you listen to psychonauts and shamans, this is well known. So what we're trying to do is go in and figure out, kind of like we figured out for sleep. Sleep isn't just sleep. Sleep has an architecture based around non-REM 1, 2, 3, 4, and then you go into what's called REM sleep, and then you come back out again. So we're trying to build, that's called the hypnogram, a trypnogram, where we can understand how these drugs are all different from each other, one. And in that, I think we're going to understand there's a lot of differential therapeutics in understanding that.
I love that. There's, I love the word trypnogram. It's a great development. And I think that's really helpful. Like we need that. And I hope scientists, I think folks are onto this. How do we hyper target for conditions and genotype, phenotype, etc.? There's so much left there.
There really is. Rochelle and I also have this other idea that, we are scientists, but I think a large proportion of this stuff has been figured out by shamans like 5,000 years ago. So if you think what a shaman did, he didn't just have somebody come into his dwelling and then hand them something and then they went away. He talked to them, figured out what was unique about them, figured out what their problem was. He understood what was happening in the village at the time. And he kind of used all that information. And really, when we said we're looking at, getting into this idea of personalized psychiatry, that's already been done. And so I really think that's where we're headed. We're going to look at everyone as an individual, look at the drugs as individuals, and then look at how they can kind of talk to each other to get mental health or, general behavior better in place.
One of my favorite lines is the shamans know what clients to pick. So they have better performance.
For sure.
There's another one, just to not be as glib. A lot of the methods they're using when we take a neurosci lens are really rich. and we don't necessarily, like, I love to go transpersonal because that's like where I live. But like this whole thing of, oh, maybe there's a more mundane, reasonable, rational explanation for it, why that rattle works so well or why that feather works so well. It's a really, really cool conversation. I might get crucified for that, but whatever.
No, if you think about the human brain, again, a large portion of what our brain does is visual and social. So it makes sense that the interpersonal and all these things are critical. And if you look at therapy, right, it's really the relationship with the therapist that's critical in some of the outcomes. So again, I think as neuroscientists, we need to start to listen to therapists more. We need to listen to shamans more, and even people into the spiritual aspects of this. They're using all the same language. It's just a different verbiage or a different way of saying the same thing.
Love that.
I also agree that I think we need to give the shamans more credit. And this is, again, another spot where Dustin and I took inspiration. They were actually using pretty highly curated combinations of different compounds, like they weren't just stumbling blankly in the dark. They were carefully selecting certain compounds to be used in combination with other compounds, in very specific circumstances. And some of those combinations of compounds are thought to enhance the entheogenic effects and peak subjective experience, if you prefer one term over the other. And we already know that if you have a good entheogenic experience, then your potential therapeutic benefit is higher. And so, they already knew some of these compounds that could be added into the psychedelic practice to enhance that benefit. And so, I think that's again a whole nother spot where at Tesselate we've tried to think about, what knowledge can we pull from there? What are they using? And even contemporary psychedelic practices often involve stacking. So stacking is, adding on lion's mane when you use psilocybin or something like that. What is in lion's mane? Why is it effective? Why have people selected it? Again, they're not just like wandering through the grocery store thinking, I should grab this mushroom because it looks cool. They're doing it for some purpose, right? Because it enhances the benefit in some way. So understanding things like that. Why did shamans add other types of compounds like wild tobacco, for example? And why do people stack? Why are we adding these things in? What's in them?
I think the more mature thing to do here is respond with curiosity, as opposed to go, "Oh, that's stupid. Of course they're wrong and dumb." And
Absolutely, 100%.
one of my
100%.
favorite comments on like traditional Chinese medicine is in the way that developed versus how we developed medicine in the West is we were like dissecting a lot of dead stuff. We're like, oh, whatever, it's just all hydraulics in there, fine. And then in China, they were systematically studying people for hundreds of years, saying, oh, look, we've got this big empire here. We can have like an institution to help us develop this, and cool, like we can all share our data. And they ended up with a really interesting thing and a really interesting model. I don't really know what to do with that model these days. I like it. I don't know how to relate to it, but I'm glad it's out there, and I'm glad there was a system that developed and is kind of like a complete system in and of itself. That's wildly different from our model. Yeah. So, is the tryptamine space too crowded?
It seems like we could get in trouble for that.
Well,
but,
some businesses went away recently. we'll put it that way. Like, so
Yeah.
you're looking at kind of non-tryptamine psychedelics. Are you exclusively looking at phenethylamines, or where are you living?
Yeah, in terms of our psychedelics, we're exclusively looking at phenethylamines, and we're really looking at synthetics. We understand that mescaline is an incredible compound. It's really revered by people that have used it, but mescaline comes from very slow-growing cactus, and probably is not going to be sustainable unless we really figure something out quick. And so, in order for us to access those types of compounds, we have gone really the synthetic route, looking at phenethylamines with some similarities to mescaline, but again, sort of in a different camp. And again, we definitely thought that looking at some of the other classes of psychedelics might be very fruitful because, again, they're fundamentally different in terms of, how they activate these patterns of brain activity and also when the plasticity is occurring afterward and, some other subtleties that we've been looking at. So.
And one of the other amazing things is back to your astute comment on curiosity. We went in trying to find, 4 or 5 hits, psychedelic compounds. And it turned out our first library, they are all hits. They are all psychedelic. But within that, some of them are like wildly short and intense, almost like DMT. And some are like low level, long for 2 days. So back to just use mescaline. Well, kind of building on that, we found out very different molecules that have very different effects from each other, which we think are going to be instrumental in treating different people with different disorders.
On a really short-acting substance that would have a trajectory like DMT, what kind of disorders would you be hopeful for, like them being helpful for? And this is all speculative. We're not tying you to this opinion.
So one of my biggest heroes in mental health is somebody called Gabor Mate, and he just released a book, and I get absolutely no money if you read it, but I suggest you read it. It's an amazing book. And he has one of these hypotheses that most of our problems, whether they're addiction or depression, they come from childhood trauma, something that was missing. So I really think, as Rochelle said in her expertise, that we don't really understand how the cortex develops over time. Trauma during that development can alter those connections and then create all kinds of future problems. So, I really think that anything that's going to rewire the cortex, and DMT is definitely one of those, is going to be helpful at kind of these trauma type syndromes. Also to that, we have evidence with compounds too, that maybe things like traumatic brain injury, where the injury is devastating the motor cortex or places like that, we can give psychedelics and kind of help rewire. Same with stroke. And then we don't have any research on that, but I know there is some research out there looking at even Alzheimer's disease. So really, I think what's happening here is anywhere where you need to change or alter synapses, psychedelics are really good at doing that.
And again, what we've been seeing in our own basic research is even if the experience is short, there is still plasticity that follows because, during the experience, that's actually not when the brain is really rewiring. Rewiring the brain actually takes a lot of time. Synapses, they're structures. It's like, remodeling a home. It doesn't just happen, in an instant. And so you need to make new proteins. You need to change, the membrane. You need to get different types of machinery there. It requires energy. And so while psychedelics really seem to be triggering these changes, a lot of time is required. So maybe that trigger is going to be enough. And then we still get the plasticity that happens downstream in the hours to days that follow. But all we needed to do was really just initiate that activity. And again, that's hypothetical. We don't have enough data to say for sure whether that's the case. And we're looking pretty carefully at the plasticity that's induced by different psychedelics. And, is there differences in magnitude or character of the type of plasticity that's happening downstream of different types of psychedelic experiences?
And I think related to this plasticity, kind of a basic science piece about the brain that a lot of people aren't aware of is that the brain is a cholesterol miser. Cholesterol is important because it makes all the cell membranes and it makes all the new synapses. There's no way past development of getting more or less cholesterol in. So really what these compounds are doing is allowing the cells to reorganize their cholesterol, to change their connections. And it's kind of beautiful if you think about it. It's kind of like returning us to a childhood period so we can see the world in a different veil or reality again.
Horrible question, but have
Those
there
are my
been any
favorite.
research done on injecting cholesterol into the brain layer on animals? Like, does that do anything?
There's something interesting about the brain, and it's not like a piece of bubble wrap. It's something called the blood-brain barrier, and it's everywhere in your brain. It's on every vessel, and it does a really good job of keeping things out and selecting very precisely what to put in. So there has been some research injecting direct cholesterol into the brain, but there's then a problem of getting where it needs to be. I'm not super up on that research, truthfully, but I know for plasticity, there's a lot of molecules in play related to cholesterol that you need to make new spines.
In regards to like the short acting substance that you talked about earlier, like I've thought about this for a while around TBI, and apparently I got multiple a year somehow, but extreme sports, yeah. So I'm thinking like a microdose of that, like smoked, inhaled, whatever, just getting it in there regularly. The person doesn't necessarily need to have that crazy experience, but a sub-perceptual experience of a short-acting thing over time, healing the brain. I would love to see some human trials on that.
That would be super cool. The one caveat I would put to that is, again, some of the research that we have in the lab looking at electroconvulsive shock. Now, back to nobody wants to take psychedelics because they don't want to throw up. Nobody really wants to get electrocuted. So there's a lot of stigma around ECS. It is highly effective, however. And just part of that effectiveness that
the short-term memory thing overblown then?
No, absolutely a problem.
No,
absolutely
that's a huge problem.
But again, go back to that cholesterol comment. If you have cholesterol somewhere for spines, right, and now we take that away and put it in something that helps you with mood, yeah, something has to go, right? And it's short-term memory. But the idea here is that with TBI and ECS, I think you're going to need a significant dose at the right time. And so obviously just following your TBI, it's probably not a good time to do it. We don't even have that figured out yet. What is the good window to give drugs for people following TBI? There was a whole bunch of studies and drugs that came out of the late 90s and the early 2000s relating to glutamate. And half of these compounds were really effective in some people, and they were deleterious in others. And so glutamate is a great way to make new connections and also to kill cells. So again, we need to understand the context, right? Set and setting in the brain itself, if you will.
Again, basic science. We have a lot to fill in here.
Job security.
As neuroscientists, how does it feel like? So we kind of got into it earlier. I love the I think it was Alan Watts making that kind of like telescope microscope analogy. Like we finally have a microscope / telescope for the brain or for the psyche. Like how revolutionary did it feel? Like, we kind of got into that a little bit earlier, but was it just like, oh, look, we can finally understand these receptor sites better. It's more obvious. Or how did that go for you both?
Yeah, I think it was truly revolutionary. And I think for me, I was a bit of a born skeptic, because I think for me, I was like, this can't be right. This can't be true. And, it just seems so off the scale. And I think that, again, being a bit of a skeptic, a bit of a pessimist, we actually tried hard to think about ways that it was going wrong, or things that, we didn't do right, or things that we should try differently, to push the paradigms more, or maybe we should add more animals, or, what could be going on. I think that was, again, for me, that was almost the feeling like this is too good to be true in terms of our preclinical results, with some of these well-known psychedelics. So what's wrong with the picture? So I guess that was sort of a feeling that I had in the beginning.
And for me, it was kind of similar. Again, as a skeptic, but as the data rolled in, you can't deny it. You absolutely can't deny it. You can have people that have no idea, trainees, running these experiments for you with no expectations, and it comes out the exact same way. So, I almost felt, to be honest, Joe, I felt cheated. I'm like, why don't we know about this? Like, why doesn't everyone know about this? And, at some level, as we've gotten into it, even our colleagues, and we mostly get jokes, right? It's, why are you studying this? Oh, consciousness and psychedelics. But when I show them the hard data, there's something called significance in science. When you have graphs, one group is different than the other. They look at our significance and they're like, wow, that is impressive. They get it. So it's been an amazing voyage so far. But, there's definitely tribes, right? And that's one of the big things we've been trying to, navigate.
It's going to be frustrating, but also a fascinating project. Like I'm sure at the beginning, people, even after showing them the data, they're like, yeah, right, sure. They're, the people over at Hopkins are high, whatever. Like, don't believe it. But like over time, and as pop attention has come to it, I bet has it become more easily sellable to your colleagues?
I'm not sure. Let's start with that. But at least we're able to kind of, release some of the stigma and work on some of the facts. So probably one of the most common things that we get in our conversations with colleagues, and truthfully, even with some VC, is that, are these drugs addictive? So everyone knows, about fentanyl and OxyContin. Everyone's really worried about that. And so when we tell them, in fact, we have some beautiful data in our lab showing in models of alcohol use disorder, they cure addiction. They're really good. I think it opens eyes, but again, people are skeptics. So, I think that's a large portion of our job, is to help educate people that these are good. And, we have conversations the other way too, where, somebody's telling me that, psilocybin cures everything from the common cold to whatever it is. And I have to say, look, there's problems here. We need to figure it all out and be sure. So again, that's what I say when I think we're kind of in between tribes sometimes.
Beyond tryptamines being a little crowded, like what's unique about phenethylamines that, most people are they're fairly familiar with LSD, DMT, psilocybin, etc. But mescaline these days, wildly rare in the underground markets. Like, it's a little expensive to produce synthetically from what I understand, but 2CB coming around a lot more. Is MDMA a phenethylamine? I believe yes, but I'm not 100% on that. Ish.
I'm
No.
I'm truthfully not 100% because I know it has other effects that
Yeah.
you don't expect as
Right.
well.
Yeah. Like it's not like I think of mescaline as like the archetypal phenethylamine.
Prototypical, yeah.
Yes.
Yeah.
Yeah. According to Wiki, yes, it is. MDMA is in the category,
There we
but
go.
yeah, like effect wise, it's different, right? Like,
Yeah.
and
Yeah.
that title's more about structure, right?
Absolutely, structure and also specificity, right? So these are going to interact with the receptors at kind of different levels. Back to the psilocybin for everyone. Psilocybin is not that effective at alcohol use disorder. There are some trials where it has shown some efficacy, but we've known for years that mescaline is highly effective. So that's another impetus for us
Based on
is to
anecdotal stuff from NAC, or like how do you get that?
So yes, from NAC, and then there's actually a couple trials in South America that have looked at for it, and more importantly, we've done the research in our own lab, so we've been able to see
There you go.
the change. And actually, we have some cool research right now looking at alcohol consumption following PTSD, so a model of PTSD, and it's really good at ameliorating that also. So I think I don't have the answer. I don't know why. We were targeting certain receptors, and we were targeting certain circuitry that we knew this would be highly important in modulating, and a compound that would work with our modulators also. And I have no idea why not a lot of others are looking at it. So we feel fortunate, and we think it is a great group of molecules. These are, again, back to, you said TCB, these are some of Shulgin's favorite compounds, right? So I think we're wise to listen to him and see what he liked and enjoyed.
Mm. Have you had any interactions yet with this gentleman, Dr. Z? Have you heard of this gentleman?
I have. Let's just say I've heard him. I have not interacted, but
I'm going to
I'd
record
love
with him
to
next
visit
week.
that.
Fascinating character. I think he's doing a lot of legal highs work, and neo-Shulginist is how he categorizes himself. And
Very cool.
he's
Awesome.
he's got the whole framework. He's got all the manufacture. It's fascinating.
Yeah, look
Claims
forward to listening to that.
Can't wait. He's never broken a law. I'm like, oh, yeah, sure, but I'm very interested to learn more because that's just endlessly interesting. The fact that somebody's willing to do self-experimentation, as Shulgin did.
Yeah, we have some colleagues that talk about that. They say sometimes that they used the compound on a large mammal. That's not our stick, but yes, that definitely exists out there.
Tested on a large mammal. It's like the internet equivalent of like someone I know, someone I met. I'm like, okay. Yeah, but it's cool. It's nice to be able to have those pieces of cover. But it's and it's also really nice to have somebody who's, you could kind of call him a researcher, also
For sure.
kind of a business person, but he's trying to put the data out there. He's got a whole business framework.
Trying to do things in a systematic way, I think is really important. And I do think that, anyone can do research, and really being systematic and trying to be detailed and careful is really what it takes. And that, that deep knowledge is really what's going to help increase the access and the acceptability. So I think more information, better.
Yeah, and maybe a dark secret that I'll share as long as you don't tell anyone is that when we were starting to use some of the NBOs, there's no information out there on dose. So we went to Reddit and we saw what people were using for dose just to start, and then we figured out a dose curve from there. But I think, again, back to curiosity, if you want the answer, you should listen to everyone. You should be skeptic, but you should listen to everyone.
Yeah, synthesize an answer based on a huge amount of research. Totally. And that's one of the most intellectually honest ways to approach it, because you get pigeonholed if you just follow, cult leader A or B, or tribal leader A or B, right? Yeah, it's really important. So what does like your timeline look like? So you've done a bunch of animal work. Like, how soon are people going to be able to consume these interesting compounds you've got?
Well, right now we're doing our initial raise, and as soon as we get this money, what we're hoping to do is then move into clinical one, and we think it's going to go fast. We have aspirations for that. We have some people on our board who have a lot of expertise in this. And again, we have lots of contacts from our farmer days at trying to figure out how to get this through. So, optimistically, I'd say a year or two, but I'm not sure about anything in the world these days. So,
Fair.
um,
Yeah, that's interesting. And obviously, first would have to be some sort of like human safety study. Is that like 1 to 4
yeah,
people or something?
we still have to do some IND enabling work. And then, from there, we'll be able to move to phase 1, which again, phase 1 generally looks mostly at tolerability. Um, Phase 1 is often not even looking so much at beneficial outcomes. We're just trying to figure out, does it not do anything too bad to people?
and a lot of our modulators kind of are some chemicals that have been used in kind of other applications. So usually and surprisingly other applications, things that you wouldn't really predict. And so one of the benefits of that is that you're able to kind of streamline the process because a lot of this work has been done already.
Yeah, it's interesting. And I guess finally, like how hopeful are you for this whole movement to revolutionize healthcare generally?
1,000%.
1,000, yeah,
So hopeful.
Fully
yeah,
in.
Yeah.
Yeah. And I'm not at the stage where I think this is our last hope. I think there's other
no,
hopes, but
it's the next
I
next
really
next
see
best
a
hope, right?
revolution. Yeah, I see a revolution here. I really do, because it's impacting the basic science that we're doing also. So people need this. People deserve it. And again, back to our vets. That's one group that it always breaks my heart. We have lots of conversations with them, and here's people that have PTSD and are having to use their own money to go to another country to get treatment that's highly effective than we know we are. I mean, morality wise, that has to change. So I'm super hopeful that people will see that, and we'll move forward into a new age where we see these as the rightful place. Things that are going to heal us. And I'm also actually hopeful that someday you'll be able to maybe take a compound like this once a year, should you choose to, just to broaden your own horizons. But that's a little ways away.
Yeah, yeah. Right now, you have to be an outlaw.
Yeah,
Super
isn't
fun.
that terrible?
So, there's a few other things I wanted to ask here. Is there, from your perspective, any way that some things would happen in the wild where the feds would try to shut down research again? I think we're at a point in
We have a
science where they
really
can't turn it around.
So I don't know is the answer to that
Yeah.
quickly. I think that we have a good relationship with the local DA. And then for us, with a lot of our research, because the amounts that we're using are so small, like what we need for a year in the lab for mice is not enough for a single human. Um, I don't see it happening. And again, we were in a conversation a couple of days ago with the DA and a group of others regarding some legislation in Nevada, and they're very supportive. So I don't know is the answer. I'm aware of what happened in Washington, D.C. with some people. I'm aware of people not being able to get meds for their patients. So I don't know, but I hope we can move forward.
Yeah, and we've really put effort into trying to build a relationship and working hard to ensure that we have amazing record keeping and we have amazing sites that are very well set up and very well controlled. They do a site visit. We make sure that everything is top notch and view them as people who are employed to do this job. It's not like we think of them as some like evil faceless entity, right? These are people doing a job, and we've built a relationship with them that is based on us doing the work that we're doing appropriately, with the right measures in place, in a controlled fashion, in a responsible fashion. So I do think that again, it's all these perspectives, all the different players coming together is going to be what's required to actually move this whole enterprise forward. Because if you don't have law enforcement and the DEA on board when you change legislation in a county or district or state or whatever, it's going to make for a lot of unnecessary challenges. And I think, again, we really are in favor of having everybody involved in these conversations, and everybody, contributing to the advancement of knowledge and the understanding of how things should move forward.
I love that. Yeah. I've yet to spend a day knowingly with a DEA employee. Someday. Meaning maybe I've talked to some and not known it. Yeah. So anyway, I think that's good for right now. Like, I'm really hopeful for your project here. It's super exciting. I love that you're on a whole different category of substance from the majority of the folks out there. And just what you said about having so much psychoactivity in the drugs you selected at first, like that alone is unbelievable. And I'm really excited to see what happens there. Not even psychoactivity, psychedelic activity. Is there a term yet for that?
I don't know. Maybe you just coined it.
Psychedelic activity. Yeah, let's use that. I'll let you publish it, and I won't require too much credit. Cool. Well, any closing thoughts, or how should people reach out to you if they care to, or follow up with your website?
Yeah, Dustin at tesselate.ai, easy enough, or Rochelle at tesselate.ai is easy enough to reach out to us at.
Yeah, we've really been enjoying conversations that we've been having with people, and also yourself. Thank you so much for having us. This has really been delightful.
Yeah, so much fun. Thank you both, and I hope next time it's in person in Las Vegas or elsewhere.
All awesome. Right. We'll go climbing.
Oh, hell yeah. All right. Thank you both.